Okay, hi, welcome back. So we're going to pick up here talking about the gene pool. So the gene pool is all of the alleles of all of the genes existing in all of the individuals of a population. So it's the entirety of the genes for a population. So let's just write that down. Gene pool. All alleles of all genes of all population. And so for evolution to occur, we have to have changes to the gene pool. So here we have an example of a hypothetical population with each oval representing a different individual organism. And we've got three different alleles in the population. We're calling one X1, which is the yellow, X2, which is the red, and X3, which is the blue. And these alleles exist at a locus, so that is, remember, that is the location on a chromosome where a gene is found. So we're looking at diploid individuals, so each has, since they have two of each chromosome, they have two alleles. This one is homozygous for the X1 gene, this one's heterozygous, etc. And so when we're measuring, evolution, basically what we're just looking at is how the proportions of these different alleles change in the population over time. And sometimes we refer to this as microevolution, which is evolution at the smallest scale possible to measure it, which is the population level. Later in the semester, we are going to talk about macroevolution. So this is large scale, I can't spell today, sorry. It's large scale evolutionary change. So examples would be things like the evolution of entire groups of organisms, or the evolution of flight, something that is occurring sort of above just the allele frequency in a population level. So, but we will talk more about this later on. Right now we're really focusing at this population level as we go through examples today. But keep in mind that it is, of course, the progression of these smaller, what you might call microevolutionary changes, that eventually give us our large-scale macroevolutionary changes, right? So it's all the same mechanism. It's just how much time and how dramatic has the change been. Okay, so evolution requires genetic variation. So you gotta have genetic variation. So we can observe some genetic variation here. So this is just differences among individuals in a population in the composition of their genes. Which is what is underlying the phenotypic variation we're seeing here in the different colors we see in the horse's fur, the different beak sizes and shapes we're seeing here, the different morphology and colors of the flower. These are all examples of genetic variation. And we have to have genetic variation for evolution to occur, because otherwise there is no genotype that's going to be more successful. Everybody's the same. So this is a prerequisite of evolution. Now, there are 5 underlying mechanisms of evolution. And we already talked about one, because we talked about natural selection. That is the one Darwin identified. But there are some other ones we're going to talk about. So first, mutation. And I'm just going to list them for now. Second, natural selection. Third, something called genetic drift. Fourth, something called gene flow, and I, of course, will be talking in more detail about each of these. And 5, sexual selection. Okay, so these are the 5 mechanisms that can cause evolutionary change, that can cause a change in allele frequency in a population from one generation to the next. Kind of a little asterisk here. Natural selection is the only one of these mechanisms that leads to an improvement in terms of the matching of an organism to their environment. This is the only, what we would call, adaptive mechanism. And I'm also going to put a little asterisk here next to sexual selection, because sexual selection is really sort of a subtype of natural selection. So this is sort of a we'll call it a subtype. I don't know if that's a real word, but I think you get my drift. Subtype of natural selection. Okay. So let's talk a little bit about these mechanisms. So first, mutation. Okay, so mutation is, and this is important, the ultimate source of genetic variation. This is where it all starts. So, most of you know what mutation is, but little refresher, this is a random change typically in the nucleotide sequence of an individual's DNA, which is what we're seeing up in this figure here. We've got our original DNA sequence, and then we've got what's called a point mutation. If you took 1406 with me, we talked about this. And again, as always, if you ever need a refresher on some of these things, let me know, and I will do what I can to help. Interestingly, you can also get kind of a large-scale mutation known as a gene duplication, which is what we're seeing here with this chromosome. What can happen sometimes is that actually an entire piece of chromosome can get duplicated, which can lead to duplication of the amount of proteins made. So that can also be a source of genetic variation. As of course can, something like a point mutation. You change the nucleotide sequence and you change the protein and you can alter its function. And usually these are bad, right? Most of us know that typically a mutation is not something that you want, but occasionally it confers a benefit. And when it does, if it does, natural selection can increase its frequency. So we also get variation, I think it's worth noting, from sexual reproduction. So again, variation is important for evolution to occur, so we can also get variation from sexual reproduction. Now this does not create new genes. It cannot generate new alleles, but it can generate novel combinations of alleles that natural selection can act on. Right, so if you think of each gene as a card in a deck of cards, now you can't sexual reproduction can't give you a new card, which mutation can do, but sexual reproduction can reshuffle the deck. And sometimes a novel combination of alleles is something that can be selected for. Okay, so natural selection. I won't talk much here because we already have discussed this a bit, but let's talk about it a little. Natural selection. So this was the one that Darwin is famous for talking about. So here we have in natural selection the differential success of individuals possessing or lacking a particular trait. So in this example, we have beetles, and the trait in question is the color of the shell, and in this case, individuals that possess the trait of having the orange shell instead of the green shell are benefiting because the predators are not seeing them, and therefore it is those individuals that are going to pass their genes on to the next generation because they will have higher survival and reproductive success because of this trait. So higher survival so individuals that have higher survival and reproductive success due to a particular trait compared to those without the trait. And it's worth noting that the thing that's important about survival is that you need to survive in order to reproduce. Because really, reproducing is what counts here. Reproducing is what matters in terms of being successful and passing your trait on. Okay, so genetic drift. Let's talk about genetic drift. So genetic drift occurs when chance events cause allele frequency to fluctuate among generations. So, and that it is a chance event is really important. So a chance event that causes, again, I can't write today apparently, change in allele frequency among generations. So there's no driving force here. It's just random chance. And this is especially something that is especially an issue in small populations. And I like this example in this picture. So let's say we have a very small population of these beetles. And, less than 10 individuals. And a random chance, in this case, the random chance is somebody stepping on them, right? Random chance we step on those beetles. And so in the next generation, allele frequency has changed pretty dramatically. And this, brown phenotype and genotype is much more common. Now, what makes this genetic drift is that these beetles not getting stepped on had nothing to do with their genotype, okay? It's not like somehow their trait kept them from being stepped on. That's what makes it genetic drift. It's a random occurrence that has a dramatic change in the genotype frequency in the next generation. And again, this is going to be most likely in, again, very small populations because it's basically like sampling error. Okay, so there are two kind of circumstances of genetic drift that are really common. We see a lot, and particularly, again, in small populations. And the first is called the founder effect. So this occurs when a new population is established by a small number of migrants. So we have a new population by a small number of migrants, and they are not representative of the source population at large. So once they migrate and they're cut off from the former population, so we don't have the back and forth of alleles anymore, the new population is pretty unbalanced in terms of allele, and we get, sort of a rapid, in this case we're looking again, I guess I have a lot of beetle examples. We have a source population that has a relatively equal distribution of color genotypes, and but we have one or maybe two individuals migrating to this island, and here we see that genotype, that red genotype, is way overrepresented compared to the source population. And we can even, in this case, get what we call fixation, which is when a particular allele rises to 100%, so everybody has it, or it can be fixed to 0%, so nobody has it. And once that happens, we call it fixation because you're stuck there, unless a mutation or more likely migration brings that allele back. Something that's somewhat similar to the founder effect is what we call the bottleneck effect. So the bottleneck effect is where you get a severe drop in population size due to a sudden environmental change. So some kind of massive drop in population size, typically due to some kind of environmental change that is sudden, something like fire, flood, earthquake. And if you think of the alleles as being marbles in a bottle, imagine if you shook that bottle, just by random chance, what passes through that bottleneck is a smaller subsample of the alleles present at large. So again, in the surviving population, you have this non-representative sample coming out. And so one example are cheetahs. So cheetahs have gone through several population bottlenecks, and their genetic diversity as a result is very limited. And in fact, any cheetah can, I believe, accept a skin graft from any other cheetah, which, I guess is good if you're a cheetah with a bad burn, but it's because there's so little genetic diversity, and that is not a good thing. All right, gene flow. Moving right along. So gene flow, also sometimes just called migration. So that's the other less fancy word for it. So this is the transfer of alleles in or out of a population. Transfer of or out of a population by fertile individuals or their gametes. For example, if we're talking about a population of flowers, maybe pollen from another population is blown over there, so it doesn't necessarily have to be the animal or organism itself. Depends what kind of organism you mean, but a lot of times we think of this as, hey, this little brown beetle, walks its way over to another population, mates, and is spreading its allele in. So it's almost the opposite of genetic drift, really. We're adding in more variation. So this increases variation. And then finally, sexual selection, which, as I said earlier, is really a type of natural selection. So this is where individuals with certain inherited characters are more likely to obtain mates. And therefore have higher reproductive success. So we can see this in a couple, oops, I'm ahead of myself here. There's a couple types of sexual selection. There's what we call intersexual selection. This is also sometimes just called mate choice. So this is where the female, usually it's the female that's the choosy sex, selects an individual of the opposite sex, in this case males, based on traits that she thinks make him look like a good potential mate. So in this case, with the peacocks, the female peacock, as you can see, is nowhere near as brightly ornamented as the male, and that is because she is the one making the choice, and it is the male who needs to go about growing these big, huge, elaborate displays to try to get mating with the female. So that is intersexual selection. Now, intrasexual selection is most often seen in competition between members of the same sex. Competition between, that's supposed to be a B, members of same gender. So, for example, two bucks that are fighting each other for access to the females. So this can, make, the winner have access to more mates. So there are some drawbacks to sexual selection. I mean, the obvious benefit to the organism doing it is they get to mate and they pass their genes on, and that is what keeps the trait proliferating and proliferating. Oftentimes these traits that are sexually selected for involve very conspicuous characters, such as very bright colors, behaviors that make them very conspicuous. And while these may attract a mate, it actually can be detrimental to the one that possesses it, because how well can you really fly and get away from a predator when your tail is this long? Also, you're really easy to see, So there are some very obvious drawbacks to sexual selection, mostly due to higher predation rates, but also just the energy put in to developing the trait. So, that takes a lot of energy to grow those feathers and keep them, looking nice. But these traits proliferate because the advantage of reproduction outweighs the disadvantage to reduce survival, so they keep proliferating. Okay, I'm going to take a break here, and I apologize I keep sniffling. I have a little cold, but I'm okay. Alright, I will see you in the next video.